Hello. The video you're about to watch is part of the Age Presents video series. This series is produced by the American Aging Association to provide an introduction to foundational and advanced topics in geroscience research. As you proceed through these videos, we encourage you to think about the following questions. 1. What relationships between aging and disease are described in the video? 2. What anti-aging interventions are proposed? And 3, what model systems were used in the research and why? We would like to thank our sponsors, the University of Washington Healthy Aging and Longevity Research Institute, the Jackson Laboratory Center for Aging Research, and the Nathan Schock Centers of Excellence at both of these institutions. If you are not yet a member, we encourage you to visit our website and become a member of AGE. AGE members receive free access to our Society Journal Geroscience, reduced membership fees for our annual scientific meetings, opportunities to interact with leaders in the field, all of the lecture materials and slides for this lecture series, and much more. In addition, we have a highly active trainee chapter, which provides numerous opportunities for networking and career development. On behalf of AGE, thank you for watching and enjoy the lecture.
Hello, my name is Laura Niedernhofer, and I'm at the University of Minnesota, where I'm a professor of biochemistry, molecular biology, and biophysics. I also direct an institute on the biology of aging and metabolism, and my lecture is going to be on genome instability and its role in aging. So let's start with the definition of genome instability. I think of it as any change that occurs to the DNA or our genetic code. And in particular, my research is focused on the nuclear genome. So of course, lots of changes can happen to that genome. There can be changes in the DNA sequence, and this is caused by chemical instability. It turns out that DNA is unstable at body temperature and pH in an oxygen-rich environment. So hydrolysis of the DNA bases occurs quite frequently, as does deamination. And as you can imagine, this is going to change the coding properties, which will lead to, upon replication, mutations. And there's many types of mutations that occur: transitions, which is a switch from one purine to another, or transversions, which would be a switch from a purine to a pyrimidine. There can also be insertions of nucleotides into the nuclear genome and deletions of nucleotides. And in areas of sequence that are particularly difficult to replicate, there can be expansions. So you get repetitive elements expanded during replication. Of course, other problems occur too. So the backbone of the DNA can break. And these breaks can be either single strand breaks, where just one of the phosphate backbones is broken. And there's also double strand breaks. And these can be either one-ended, say with what occurs during replication stress when the replication fork is broken, or a two-ended double strand break where a chromosome is broken into two pieces. There's also epigenetic changes to our genome, and these are purposeful to regulate expression of genes. However, these epigenetic changes are sometimes unstable and can also attract enzymes that modify them or attract DNA repair proteins as well, and this can lead to trouble. And finally, the telomeric DNA at the ends of our chromosomes turns out to be particularly vulnerable to instability. And that's because as we replicate our genome to create daughter cells, there's an attrition of this telomeric DNA with every round of replication. And it turns out that telomeric sequences are very vulnerable to chemical instability, modification, as well as strand breaks. So what drives genome instability? Well, here's some common sources of DNA damage. They can be environmental, endogenous, or even therapeutic. So in our environment, some examples of genotoxic stress that drives genomic instability is the UV component of sunlight, which potently modifies DNA, also cigarette smoke, and even polyaromatic hydrocarbons, which are just superheated carbon molecules. This occurs in air pollution, but also when we grill outside at high temperatures, some of that fat around your steak is actually going to contain some chemicals that can modify DNA. Endogenously, our mitochondria produce reactive oxygen species that can react with DNA. And we also alkylate molecules purposely in the body as part of normal metabolism, but alkylation damage can also occur in the genome. And then there are many, many therapeutics, particularly used in oncology, that purposely damage DNA and lead to genome instability. What's really interesting to look at is the amount of DNA damage you get either endogenously, which is shown on top, or through environmental exposures. So if we look at the frequency of these various types of DNA lesions per cell per day, you get about 10 double-strand breaks, about 100 deamination of cytosine residues. Cyclopurine addicts are oxidative DNA lesions. They occur at about 100 times per cell per day. Depyrimidation or depurination can occur on the order of tens of thousands of lesions per day, as do single-strand breaks. Adeoxo-G, as well as malondialdehyde addicts, are also oxidative DNA lesions. So that you can see that each cell in your body experiences tens of thousands of lesions per day that it must deal with to avoid genome instability. By comparison, if you look at these environmental exposures, sunlight will cause photodimers, but only about 100 of them in the skin, and the UV does not penetrate deeper than that. Background radiation can lead to single-strand breaks, double-strand breaks, but they're relatively infrequent compared to what occurs endogenously. And then we have ionizing radiation or cancer therapy with chemotherapeutic agents, and they lead to very large amounts of DNA lesions, say on the order of 1,000 per cell. And that's with the intention of killing those cells to stop tumor growth. So for each type of DNA damage, there are very robust and highly conserved DNA repair mechanisms. So DNA is really the only biomolecule in our bodies that's actively repaired. It can't just be chewed up and replaced. So we have these very highly conserved mechanisms of repair. So there's nucleotide excision repair that will recognize and remove bulky changes to DNA bases. They have to be big enough to distort the DNA helix before they're detected by nucleotide excision repair. Interstrand crosslinks are lesions that tether the two strands of DNA together. You can imagine that these can be very harmful because it prevents DNA replication as well as transcription. And these interstrand crosslinks have a distinct pathway for their repair. Very small base changes are repaired by what's called base excision repair. So this just cuts out a single nucleotide or just a couple of bases to replace these. This base excision repair is absolutely essential for life because of the high burden of endogenous DNA lesions that occur in our genome, like adeoxo-DG that's depicted here. And then for double-strand breaks, we have homologous recombination or non-homologous end joining, which occurs if the two ends of a double-strand break are easily ligated together. So it would take quite a bit of time to go through the mechanisms of repair individually. So I'm going to use one as just an example. So I'm going to illustrate for you nucleotide excision repair. And again, the lesion has to be bulky enough to perturb the DNA helix. And this will attract a complex of proteins depicted with these little bubbles that will detect helical distortion and land on it. Depending on the type of lesion causing the distortion, there might be other accessory proteins that help with rapid detection of the addict. And the other way you could detect DNA lesions that will trigger nucleotide excision repair is if you're trying to transcribe the DNA. So RNA pol 2 is coming along and it crashes into the DNA addict and cannot finish its job. So after this, the repair pathway converges. So once you've detected a DNA lesion, then a whole complex of proteins are attracted to that site. And this includes even a standard transcription factor, TF2H, that is used normally to open a bubble around the start site for transcription. Amongst this complex in TF2H are two helicases, XBB and XBD, so they will unwind the area around the DNA adduct. This will attract additional proteins that have to stabilize this open bubble like RPA. And then ultimately two endonucleases are attracted, XPG or the complex ERCC1 XPF. and they will incise the bubble on the damaged strand on either side of the lesion to release the damaged oligonucleotide. This leaves behind a gap that's filled by templated DNA synthesis by polymerase delta or epsilon, along with other factors, and then ultimately ligated by either ligase 1 or ligase 3. So as an illustration, these DNA repair pathways are very complex and require the choreography of a very intricate ballet to get these proteins and enzymes onto the damaged DNA in the right spot, in the right order, so that DNA damage can be excised. So what I want to point out, though, is that if you're missing this pathway entirely, it's not incompatible with life. So the disease associated with defects in nucleotide excision repair is called Xeroderma pigmentosa. And a hallmark feature of this disease is that the children born with errors in NER have a 10,000-fold increased risk of skin cancer. So you can imagine then that those UV-induced photodimers are a substrate for nucleotide excision repair. And these children are forced to wear basically NASA-like spacesuits to stay away from any UV radiation. Unfortunately, if the children are protected very well from sun exposure, they still have a very high likelihood of succumbing to neurodegeneration in the third or fourth decade of life, illustrating that there's also endogenous substrates for this DNA repair pathway. So again, while the pathways are complex, there's really three key steps to all DNA repair mechanisms. One is the DNA damage must be recognized, and there's various means to do that. Then the DNA damage must be removed, and this requires DNA endonucleases or exonucleases. And then ultimately, whatever's cut out, you'd like to replace to maintain genome stability. And so that usually involves some DNA synthesis that's templated. So what I want to do is focus for a minute on this endonuclease ERCC1-XPF, which is a platform for much of my own laboratory's research. So this was intriguing to me as a postdoctoral fellow at Jan Hoijmaker's lab in the Netherlands, because we found that if you knocked out ERCC1, it didn't give a phenotype anything like Xeroderma pigmentosa. In fact, these mice look very, very different than mice that had been deleted for other genes in the nucleotide excision repair pathway, say deletion of XPA. So deficiency of ERCC1 led to a lot of features that we thought looked an awful like accelerated aging. So here's an example where genome instability is not just driving cancer, but also an aging phenotype. And this ultimately led to the discovery of a human progeroid syndrome, or disease of accelerated aging, which was caused by mutations in XPF. So that's one of the subunits of ERCC1-XPF endonuclease. And sure enough, in these patients, you see low expression of the XPF protein, which is shown with the red arrow in the middle lane. And as a consequence, very similar to those mice, these children are born looking healthy, develop normally, but very early in life succumb to a number of age-related diseases that are quite familiar. So let me tell you a little bit more about this endonuclease. It's two subunits, ERCC1 and XPF, that bind and stabilize one another through their C-terminal helix-hairpin helix domains. And what's important to note is this stabilization is required. So if you knock out ERCC1 in mice, for instance, and then look in their tissues like liver, you cannot find XPF protein. So in vivo, these two proteins must bind, stabilize each other in order to create a functional endonuclease. And we ultimately found that this endonuclease was involved in multiple DNA repair pathways. So not just nucleotide excision repair, but also that interstrand cross-link repair pathway, and then also the end joining of some double-strand breaks. And that probably explains the very severe phenotype of these mice and the difference in how patients look when they have low expression of ERCC1-XPF compared to the disease Xeroderma pigmentosa. One other important point I'd like to make is that when we look in fractionated tissue, so take nuclear fraction versus mitochondrial, we only find these proteins in the nucleus. So we feel all of the phenotypes of accelerated aging are due to failure to protect the nuclear genome. And so giving us a hint that DNA damage or genome instability can promote aging. So we made a better mouse model of that human progeria disease. We made mice that had one knockout and one hypomorphic allele of ERCC1, and this led to animals that expressed about 5% of the normal complement of XPF. And like that patient, the mice are born with normal development in utero. They reach adulthood, which is 9 weeks of age, looking fairly healthy. But within about 5 or 6 months of life, they're really looking quite aged. So they become deaf and blind, they have urinary incontinence, they go gray, and they have this unsteady gait. So they look like they're aging about 6 times faster than normal mice. And if we don't do anything to the animals at all, we find they spontaneously develop a huge number of age-related diseases affecting the bone, the muscle, the heart, kidney, liver, lung, you name it, and these animals really have a full spectrum of age-related diseases. So we think this provides pretty darn strong evidence that DNA damage, when not fully repaired, can promote aging. And in fact, if you look at most human diseases associated with accelerated aging, there is something affected with the genome maintenance system. So in the disease Xeroderma pigmentosa, there's a defect in nucleotide excision repair, and there's accelerated aging of the skin and of the brain. Cockayne syndrome is affecting transcription-coupled nucleotide excision repair, and we see cataracts, retinal degeneration, muscle atrophy. Werner syndrome is a true accelerated aging syndrome where there's telomere problems and problems with replication, and this leads to early onset of osteoporosis, cataracts, cancer, and diabetes. So it's really interesting to note that any of the true progeroid syndromes listed in the far left column have some form of impairment in maintaining genome stability, leading to accelerated aging of one or more organ systems. Again, further evidence that DNA damage, if not well handled, can lead to aging of various organ systems. So I want to make a point that it's very, very difficult to measure endogenous DNA damage. And it's important to recognize that, again, these lesions are very abundant, yet in the total number of nucleotides in the nuclear genome, they're still relatively rare. So on average, 1 in 200,000 bases is damaged at any given time. So you have to be able to detect these lesions at femtomolar concentrations, which is tricky. These lesions are also very diverse. They could be strand breaks or various different oxidative lesions, many of which are unstable and change over time. And another problem is when you're isolating DNA from biologic samples, you can also produce DNA damage because of the oxidative environment in which we work. So the solution in the field has been to use surrogate markers or indirect measures of DNA damage. So you can see activation of the transcription factor P53, which is the guardian of the genome. So we know that it's activated whenever there's genotoxic stress. It's also true that histones that make up the nucleosomes on the chromatin can be modified in response to genotoxic stress. So we use gamma H2AX foci as a marker of DNA damage, but obviously it's a protein and not DNA. And then there's non-specific measures like the COMET assay, which it looks to see if your DNA, when put into electrophoresis under various conditions, shatters and slides out of the nuclei, leaving a tail. And the size of that tail would indicate that there's been genome instability in that particular cell. But my favorite is really this highly sensitive and specific method of using liquid chromatography, mass spectrometry, to measure DNA damage. And so this is very quantitative, it's very sensitive, and it uses an isotopically labeled internal control. So you build the adduct that you're looking for, and then isotopically label it to make sure that you're not generating DNA damage during sample preparation. So this is great, but as I just indicated, you can only do one adduct or at a time. And so it's very laborious and it's limited to the number of lesions that you're able to chemically synthesize. So we have done this in collaboration with a chemist, Yin-Sheng Wang at UC Riverside, to ask if in our DNA repair deficient mice, those ERCC1 deficient mice, do they actually have more spontaneous endogenous oxidative DNA damage? And you would expect they would. In particular, in the case of cyclopurine addicts, which are illustrated here, there's 4 of them, diastereomers of guanosine addicts as well as adenosine addicts. And these are substrates for nucleotide excision repair. So we fully expect that in our ERCC1 deficient mice, there should be higher levels of these cyclopurine addicts. I should also point out these addicts are caused by reactive oxygen species endogenously, and they lead to an addition of an extra bond between the deoxyriboside and the base so that they're very chemically stable and quite easy to measure. So here's an example from renal tissues from our mice, which are ERCC1 deficient in red bars, compared to wild type mice in blue bars. So at 10 weeks of age, you see that the mutant animals do not have more of these 4 chemical lesions. So what I've done with these histograms is it's the sum total of all 4 of the cyclopurine addicts. And again, at young age, there's no difference between mutant and wild type animals. But when the animals are older and have their accelerated aging features, there is significantly increased cyclopurine addicts in renal tissue of these animals compared to their wild type counterparts. And so this is consistent with DNA damage building up in these animals because they're unable to repair it. But what I want to point out is the bar in the far right, which is just renal tissue from old wild type mice. And you can see too that these animals have the same amount of cyclopurine addicts as the DNA repair deficient mice. It's just not until an older age. So we feel like this illustrates that DNA damage accumulates or occurs in old organisms to the same extent as it does if you wipe out DNA repair. And I think supports the idea that DNA damage could be contributing to aging, not just when you remove the repair mechanism, but even in normal physiology. What I'd like to point out too is that different tissues react very differently. So this is now comparing cyclopurine addict levels in the ERCC1 deficient mice in the orange bars compared to age-matched wild-type controls at 2 different ages, 10 weeks on the left, and at a progeroid age on the right, but in 3 different tissues, liver, kidney, and brain. So if we look on the right-hand side, there's significantly decreased amounts of cyclopurine addicts in the brain of the mutant animals compared to wild type. And that puzzled us for a while, but we think that's because when neurons in the brain undergo oxidative DNA damage, it kills them. So you actually lose the cells with the most DNA damage. In the kidney, there's this modest increase of cyclopurine addicts in the mutant animals. And we think this is because some cells die in the kidney while others may senesce or be replaced. So just a modest increase, but the liver is really striking. So there's a huge increase in cyclopurine addicts when you take away DNA repair. And we think that's because the damaged cells can actually tolerate that DNA damage. And instead of apoptosing like in the brain, they undergo cellular senescence. So they're there and they maintain their damage, unable to repair it. And therefore damage accumulates to these extraordinary levels. So I'll explain a little bit more about senescence in later slides. So overall, I think there's a number of lines of evidence that point to the fact that DNA damage could very potently drive aging. And one is these accelerated aging diseases or progerias that are seen in humans and then faithfully recapitulated in genetically engineered mice. Another thing that's becoming abundantly clear is that when we use genotoxic chemotherapy or radiation therapy to treat cancer, it leads to accelerated aging of these cancer survivors. So it's known now that a 50-year-old who is a cancer survivor has 2.5 times greater incidence of age-related diseases than a sibling that never had cancer. So this includes neurocognitive decline, cardiac disease, insulin resistance, and they become quite frail actually, and they have a decline in function, so gait speed and grip strength. So they really do functionally look like a frail older adult. So older organisms tend to have more DNA damage than younger. So this is true if endogenous oxidative lesions are measured. This is true in a number of tissues that have been reported on and in a variety of different species, mice, rats, hamsters, and even humans. But I want to point out that these are a little bit limited because they're only cross-sectional studies, and we really don't have measures of DNA damage longitudinally in individuals as they age. Another piece of evidence is that the consequences of genome instability are also increased with age. So older individuals will have more mutations and more senescent cells, which are consequences of DNA damage. And these can be chromosomal aberrations, transpositions, point mutations in a variety of different tissues in virtually every model organism or even humans that have been studied. And what's really important is you can suppress the number of mutations or senescent cells by doing anything that's an intervention that's known to extend lifespan. So for instance, caloric restriction will prevent senescence and mutations, suggesting that the way caloric restriction might work in part to extend lifespan is to suppress DNA damage. You will also find in the literature reports that decreased DNA repair capacity occurs with aging, but I would say the jury is still out on that. I don't think the evidence is particularly strong. So aging, what is it? It's very easy to recognize, but very hard to define. But Thomas Kirkwood, who is a thought leader in aging genetics for quite a number of years, stated that aging results from the accumulation of unrepaired cellular and molecular damage through evolved limitations in somatic maintenance and repair functions. And if I read that statement, it screams DNA repair to me, because there is abundance of DNA damage and we have very robust repair functions, arguing that maintaining genome stability is of utmost importance and therefore could prevent the aging process. And in the field of aging, this has led to definition of pillars or hallmarks of aging. And these are things that virtually always go wrong with aging. And they include accumulation of macromolecular damage, like DNA damage, epigenetic changes, which is also DNA damage to some extent, inflammation, which can drive DNA damage, altered adaptation to stress, like genotoxic stress, problems with proteostasis, which is harder to link to genome instability, stem cell problems, which can be because of genome instability in stem cells, and altered metabolism, which I will show you can result from too much DNA damage. But an important point about these pillars is they're all interconnected. So we began to ask questions about how is it that macromolecular damage to the genome could influence all these other pillars of aging. So in fact, what we were asking is, what is the mechanism by which DNA damage drives aging? So we have these ERCC1 deficient mice that spontaneously accumulate physiologically relevant levels and types of genotoxic stress, like those cyclopurine addicts. And what we typically think about in the world of DNA damage and repair is when you have too much DNA damage, this will lead to cell fate decisions. But this would then argue that DNA damage drives aging exclusively through a cell autonomous mechanism. So you lose functional healthy cells and eventually you would lose regenerative capacity. But in the field of aging, there's a very robust body of literature that says that there's cell non-autonomous mechanisms of aging. And the most elegant one of those, I think, is the heterochronic parabiosis studies. So this is when you stitch two animals together, an old one to a young one, and you find that within a matter of weeks, they're sharing a circulation. And what's very intriguing is that young animal is able to rejuvenate the older mouse just by sharing a circulation. And concomitantly, the older mouse is able to age the younger mouse just by sharing their blood. So this clearly illustrates the cell non-autonomous mechanisms of aging through paracrine signals, the SASP, which is a senescence-associated secretory phenotype, inflammation, metabolic changes, endocrine changes. So we wanted to explore these in the context of DNA damage. An important point, though, is if you wanted to find a way to treat aging, what you certainly would not want to do is try to inhibit these cell autonomous cell fate decisions, because these are very potent tumor suppressor mechanisms that prevent replication of a damaged genome, leading to genome instability and the possibility of cancer. Whereas cell non-autonomous mechanisms of aging could be much more druggable to slow the aging process. So again, of keen interest to try and define those. So what happens in a cell with genotoxic stress? Well, it triggers a very elegant and very complicated DNA damage response referred to as the DDR. So damage is recognized by proteins and leads to a signaling cascade that ultimately will lead to cell apoptosis, arrest of the cell cycle, to afford time to repair the DNA and recover transcription once the genome is repaired, as well as replication. So the DNA damage response includes damage sensors for each of the DNA repair pathways, as well as signal transducers, and then these effectors that lead to action or cell fate decision. And this gets enormously complicated. So this is just a very simple illustration of the DNA damage response and the key transducers, which are two kinases, ATM and ATR. And each of those has hundreds, if not thousands, of substrates. So they're phosphorylating a number of different proteins to trigger this response to DNA damage. And they also activate p53, which is a transcription factor with hundreds of transcriptional targets. So these transducers will ultimately lead to, at the bottom of the slide, apoptosis signaling, activation of DNA repair mechanisms, and a complete cell cycle arrest that's either transient or permanent. And this is what leads to maintenance of genome stability in the organism. So let's talk a little bit more about the permanent cell cycle arrest. So this is what's called cellular senescence. So I've illustrated in the bottom left here a senescent cell, and they have a number of features that are very unique. So they change their morphology. And if you look at the blow up of the nucleus, there's a number of markers of genotoxic stress. So this includes senescence-associated DNA damage foci, or SADs. There's also senescence-associated heterochromatic foci, or SAFs, gamma H2AX, which is a phosphorylated histone, and TAFs, which are telomere-associated foci. All of this occurs in a setting of an irreversible cell cycle arrest. So we see increased expression of cell cycle inhibitors like P16, P21, and their master regulator P53. There's also unusual features of the senescent cells. This SA beta gal is a senescence-associated beta-galactosidase and lysosomes of senescent cells. So the activity goes up, allowing you to identify senescent cells by administering the substrate XGAL, which will turn senescent cells blue. There's lower expressions of HMGB1 and lamin B1 and mitochondrial changes. There's cytoplasmic bits of DNA that's very inflammatory and increased reactive oxygen species and damage-associated molecular pathogens. At the very bottom, you see SAS, which was this huge secretory phenotype of senescent cells. And you can see some of the factors that are released out into their immediate environment. And you'll recognize a number of growth factors, proteases that can affect the extracellular matrix and degrade the immediate environment. There's a number of different receptors that change the identity of these cells, bioactive lipids, as well as a lot of inflammatory cytokines and chemokines. So senescent cells are thought to drive aging through this secretory phenotype by setting up an organism for a state of chronic inflammation that leads to loss of tissue homeostasis and vulnerability to virtually any stress. So senescence itself is driven by a variety of different types of cellular stress, so mitotic stress, attrition of telomere DNA, and just plain old DNA damage. And so we've been asking ourselves, with all the DNA damage that's accumulating with natural aging, as well as in our DNA repair deficient mice, do we see evidence of a lot of senescence? What I do need to point out is when you're trying to find senescent cells, As you'll probably recognize from the list of factors up here, not a single one of them is specific to senescence. So it's imperative to measure 5 or 6 of these endpoints to conclude that you have cellular senescence in tissues. So here's an example of our survey of old wild-type mice in the middle column compared to young wild-type animals. We were measuring P16 expression as one marker of cellular senescence. So this is one of those cell cycle regulators. And you see that P16 messenger RNA is strongly upregulated in aorta. And if you go down the heat map, it's sort of ranked in order of the level of P16 and where it occurs in the various tissues with normal aging. In the right column is our DNA repair deficient mice. And we were very surprised to see that we find P16 expression in the same tissues as occurs with normal aging and virtually the same levels in most tissues, certainly not exaggerated compared to normal aging. So we think this could be, while correlative, further evidence that DNA damage drives aging even when you have a normal DNA repair capacity. We also noted in our ERCC1 deficient mice, or cells derived from them, that although this enzyme is only found in the nucleus of the cell, mitochondria eventually become very sick. So in the bottom left panel is a staining of cells with diac6 to light up mitochondria, as well as mitosox, which recognizes reactive oxygen species. And when you see a lot of yellow, it's because there's too much reactive oxygen species in the mitochondria. And that's quite abundant in the ERCC1 deficient cells. On the right is some super resolution imaging, and you see that just the pattern of mitochondria is very different in the bottom. They're very punctate and fragmented. And this is what happens in senescent ERCC1 deficient cells with a lot of DNA damage. So we asked, where is this increased reactive oxygen species come from? And first of all, we measured it in liver tissue, our mutant animals. So we measured specifically superoxide anion using EPR. So the animals, the ERCC1 deficient animals are shown in red, and then compared to age-matched animals in blue to the left of them, as well as aged wild-type mice to the right. So both with normal aging and in a DNA repair deficient animals, we see increased superoxide anion. We also see damage to other molecules. So lipid peroxidation is when ROS reacts with fatty acids and membranes. So this is increased with normal aging as well as in the DNA repair deficient animals. And there's upregulation of enzymes like xanthine oxidase and NADPH oxidase, which produce reactive oxygen species enzymatically. And this occurs in the ERCC1 deficient mice as well as with normal aging. And we also see on the far right a decrease in expression and activity of potent antioxidants. So you see in the red bars that levels of catalase go down as the ERCC1 deficient mice age, as occurs with normal aging in the blue bars. And the levels of oxidized to reduced glutathione go up, meaning that there's indeed increased oxidative stress with aging in the ERCC1 deficient mice as well as normal aging. So then we next asked if this reactive oxygen species is truly pathological. So the way we approached it was to treat our animals with a reactive oxygen species scavenger that specifically targets the mitochondria. So XJB5131 is a recycling radical scavenger that intercalates into the inner mitochondrial membrane. And we were very surprised to see that when we measured those 4 cyclopurine addicts in ERCC1 deficient mice that were treated with XJB or vehicle alone, this drug actually suppressed levels of DNA damage. And I'll come back to that because I think that's really fascinating. So we had obviously less ROS and as a consequence, less oxidative DNA lesions. This was sufficient to suppress cellular senescence in the animals, whether it was measured by SA beta gal in tissue sections of the liver on the top, or by a P16 luciferase reporter that we could follow longitudinally in the animals that were treated with control in the pink dots or XJB in the blue dots. And this was also enough to suppress age-related pathologies. Here's an example of micro-CT where you see that osteoporosis, so the little holes in the vertebrae, are reduced by XJB treatment, as well as markers of neurodegeneration, like GFAP, which is a marker of gliosis in response to neurodegeneration. So that brown staining is attenuated in animals treated with XJB. So clearly, reducing ROS is beneficial to these animals, but it didn't fix DNA repair. So if we look back over at the suppressed levels of oxidative DNA lesions, what this says to me is when you have a lot of DNA damage, your mitochondria get sick, they start producing more ROS. That leads to lipid peroxidation and damage to other molecules. And it also leads to feed forward and more DNA damage. So that's the red bars with the animals treated with vehicle. So if you interrupt the cycle with a radical scavenger, you can actually improve the animals. So it means to me that once you have a certain level of DNA damage, you actually get to a state where you make more DNA damage. And this is how all of the pillars of aging kind of tie together. Although in our case, we know that we started with a DNA repair defect and DNA damage as the instigator. So we think it's ultimately this point where you start to see aging features when all the pillars of aging have been impinged upon. So no lecture on genome instability is complete without talking about telomeres. So here's a beautiful EM illustration of telomeres, and the loop is the T-loop structure. So let's dig into that a little bit more. At the very end of your chromosome, you have this G-rich strand overhang, and there's a number of proteins that make up the sheltering complex, and their job is to fold that G-rich strand 3' overhang into a T-loop. So it's literally wrapped around on it itself and intercalates so that it's displacing the normal strand of DNA, which would have been upstream of the telomere DNA, creating this displacement D loop. And the sheltering complex and all these proteins stay there to protect this structure. And this is really important because telomeres are really kind of the ends of the shoelaces. So they protect your shoelaces from becoming frayed. So telomere DNA does protect the chromosomes from shortening with each round of replication. This is a southern blot measuring specific telomere sequences, the TTA-GGG repeat that makes up telomeric DNA. And you see on the left that cells that are proliferating, they start losing length. So the darkest part of the blot is going further and further to low TRF length as cells are replicated time and time again. And eventually it just stabilizes when cells become immortal. So it turns out that chromosomes are shortened with successive generations by anywhere between 50 and 100 base pairs with each replication or passaging of cells. And ultimately, when there's less than 12 repeats left over, that's when the telomeres are so shortened, they've driven crisis, and they're recognized by the DNA damage response as a double strand break. And this will lead to either apoptosis or cellular senescence. So immortalization of cell lines that you may have used in your research requires that telomere size remains stable. And so this is usually by reintroduction of telomerase, which can then enzymatically lengthen the length of the telomere repeats. So when you hit crisis, this is what will happen to your genome. So we're looking at fluorescence in situ hybridization, or FISH, to pick up telomeric sequences. So that's what the little green dots are at the end of each of the chromosomes. And when they get critically short, basically they just fuse one to another chromosome, and you end up with this spaghetti pile that cannot be replicated or divided into daughter cells anymore. So again, if we illustrate the telomere, there's to the left non-telomeric normal DNA, and there's a subtelomeric region, and then these repeats of TTA-GGG. And then at the right-hand side, the 3' G-rich overhang. So it turns out that these runs of Gs are very hard to replicate. So telomeres are not only vulnerable to attrition with replication, but you can also have deletions or stalled replication forks, which will lead to double strand breaks. These Gs are also very vulnerable to damage, But this gap right here between double-stranded and single-stranded DNA is a substrate for endonucleases like ERCC1-XPF. All sorts of chemical electrophiles or therapeutic chemotherapy can modify these telomeric guanosine residues. And then the ends of the chromosome, when unfurled or not in the T-loop, is very vulnerable to exonucleases. So lots of ways to damage telomeres, leading to genome instability. So to wrap up, DNA damage or genotoxic stress, which drives genome instability, occurs frequently and is virtually unavoidable. Sources of genotoxic stress are the most abundant types of DNA damage that drive genome instability. All organisms have multiple, robust, and highly conserved mechanisms by which to repair their DNA damage. But unfortunately, repair is never 100% complete, or is it ever perfect? So repair can generate other problems, but also eventually DNA damage does increase in old age. There's very strong evidence to support the conclusion that genome instability contributes to aging, and DNA damage contributes to aging through cell autonomous and non-autonomous mechanisms that ultimately impact other pillars of aging. And areas of genome instability that are potentially targetable to slow the aging process would be doing what you can to avoid or prevent DNA damage. So not sunburning, not smoking, avoiding some fatty foods that are charbroiled. Also, we'd love ways to improve DNA repair, but right now I have to say that's a little bit of a lark. There just aren't chemicals that can do that or drugs, and those pathways are so complex, it's hard to rev up DNA repair endogenously. But what we can do is deal with the fates that DNA damage drives. So senescent cells are one really key target that can be druggable to prevent aging caused by DNA damage. And I just say that if you'd like to learn more, I would encourage you to look at DNA repair and mutagenesis, which is really the Bible of understanding genome stability and the consequences of DNA damage. So thank you very much for your attention.